and we'll have a live transcript running as well. So let me welcome you all today. This is the Advanced PCR series, and we had a pre-series showcase about 3 weeks ago on, let's see, October 8th. And then we had October 15th, we had our first part of the 3-part series. And that was from theory to tracking viruses in wastewater. And today we have the real-time qPCR theory and real-world assay design. And we thank Bio-Rad for helping us put this all together. And again, welcome everyone. And at this time, I request, Linnea, do you have any welcome words?
No, I just want to say, as the executive director for InnovATEBIO, I welcome all of you to this Friday, because I know how precious Fridays can be sometimes. And I thank Bio-Rad for putting this on. Thank you.
So with that, I request Bryony Rigg to please take the mic.
Thanks a lot, Purnima, and thank you very much to the InnovATEBIO Center for helping us, partnering with us to put this on. And huge thanks to Purnima and Cassandra and Linnea for working on this so closely. I'd like to introduce the 3 speakers for today: Dr. Liz Jordan, Dr. Evan Bress, and Dr. Steve Aquino. We'll start out with Liz and then go to Evan and then go to Steve. I will do each introduction just before they speak. So we're going to start with Liz. So Dr. Liz Dreskin oversees the applications team for the Life Science Group at Bio-Rad Laboratories. Her team works with the marketing team to develop data and literature to help guide scientists on how they use Bio-Rad's tools, whether that be instrumentation or reagents or software. And those tools that she works on include electrophoresis and Western blotting, PCR, quantitative PCR, droplet digital PCR, next generation sequencing, electroporation, flow cytometry, and protein purification. So thank you so much, Liz, for giving us this intro to quantitative PCR talk today.
Thanks, Bryony. Okay, let me get this started. Okay. Hi, everyone. So we'll start out with just a brief overview of PCR that we talked about last time, and then we'll move into real-time PCR and a little bit about the instrumentation, the reaction chemistries that are typically used, analysis, and then a little bit about the uses. So this is a reminder slide about PCR in general. We have the reaction components. I've got a generic Taq polymerase. I mentioned that most biotech companies have their own version of Taq polymerase and also very specialized mixes to allow us to do different types of PCR. Our reactions are hot start. The ones I'm going to refer to are always hot start, and that means that the enzyme is held inactive by either aptamers, antibodies, or chemicals until we activate it with this high temperature, 95 degrees, for 3 to 5 minutes. And then the standard cycling consists of 3 steps usually: denaturation, where the strands become disassociated, then annealing, where primers bind complement, and synthesis or extension, where the polymerase can go and extend those to create a second strand. Most of the PCR that I do actually combines steps 2 and 3 into an anneal extend step. A lot of the reagents that are available today are perfectly good working at the annealing temperature to do the extension. They're very fast. And then typical PCR reaction is 35 to 45 cycles. So what if we have RNA and we want to look at gene expression? We first have to convert that RNA into a cDNA. We do that by annealing primers such as oligo-DT, random hexamers, or gene-specific primers. And then we use a reverse transcriptase with RNase H activity, dNTPs, to go and reverse transcribe that first strand. And at the same time, destroy that original RNA copy so that we maintain fidelity with the original copies. Okay, and then we have first strand cDNA that we can use in PCR reactions. So we mentioned before, PCR is the basis for droplet digital PCR and qPCR. And we talked about conventional PCR being an endpoint readout where we either do sequencing or gel electrophoresis. Droplet digital PCR is also endpoint, but we're using fluorescence to detect the reactions, and that's very sensitive. And then quantitative PCR or real-time PCR, we're actually going to look at fluorescence at each cycle. It allows us a broader dynamic range than conventional and digital PCR. It allows us to do high throughput also. The equipment is more expensive than regular conventional PCR. And in order to be quantitative, you need to run a standard curve, which does limit some of the throughput. But let's talk about conventional PCR. So endpoint PCR, we run a PCR reaction and then we load them on an agarose gel, and here we have our bands. All we can say about these is that, yes, we amplified something. Maybe there's a loading issue here, but it looks as specific as we could tell from this gel. With real-time PCR, we're actually going to compare the fluorescence at each cycle of the whole PCR reaction and get an idea of relative starting quantities. And I'll show you a little bit more later, but if you look at this, this is the end point of these PCR reactions. And these are the same reactions. We ran them on a gel. So you can't tell anything about the initial concentrations of starting material here, but I'll show you how we can tell the difference between starting concentrations by using real-time PCR. But first, how can we detect amplification without having to run a gel? And how could we have more quantitative results than a conventional gel? So conventional PCR reactions were modified to include fluorescent detection reagents. We'll talk about those in a little bit. And instrumentation had to be designed to measure the change in fluorescence with each cycle. This is a schematic from one of Bio-Rad's instruments, but we can look at this as a generic real-time optical head, if you will, where we have a light source, excitation filter to narrow the wavelength of light that we're going to subject the sample to, some mirrors to direct the light to our sample, mirrors to detect light, to direct the light away from our sample and through an emission filter, and then some sort of a detector. The first real-time PCR experiment was reported in 1993, and this is this is what it looks like. If you were to walk into a lab today, you'll see equipment from Bio-Rad, Roche, Thermo, and QIAGEN, others too, but obviously they look a lot more modern. Can you see my pointer here?
Yeah.
Yes.
Okay,
Well,
Yes.
okay, I was just
yeah,
checking.
I could
'cause-
see it. It was
I
a
have-
little small, but yes.
Okay, good. So what happens during a PCR reaction? That initial paper in 1993 looked at a PCR reaction kind of from the viewpoint of enzymologists and what is happening during each cycle. So we can break down the sigmoidal curve into two parts. There's an exponential phase where we're doubling our products after each cycle. And then there's a plateau phase where we might be starting to run out of some of the reaction components. And they theorized that if we were to set a threshold, some like, let's say 3 to 5 fold higher than the baseline, whatever our baseline signal is, that we might be able to compare the starting material by where they cross this threshold line. And this is my real example here, where I have 10-fold dilution series. And you can compare, this is the most concentrated template. And then I've got 10-fold dilutions coming up later and later. I'll talk about that in a minute. But first we need to talk about the dyes. The original work was done with ethidium bromide and other DNA binding dyes like cybergreen and evergreen. There's no special optimization required. It's very cost effective and it's widely used today. It's non-specific binding, so it binds any double-stranded DNA that you have in your reaction, including primer dimers if they're present, including other things that might be non-specifically amplified. So we need to be careful with that. But what it allows us to do also is to do a melt curve where we, after the PCR reaction is complete, we can incrementally raise the temperature of the sample and look for an inflection point, and we call that the melt. In this case, I have a high concentration of a template and I'm getting one nice peak. It's specific. But in this case, I actually have a primer dimer problem. And so when I have very low inputs of template, the primers start to form dimers with each other. And this can interfere with your detection because it's going to be detected just as much as the real product that we're looking for. And I'll get to a point, I'll show you how that can interfere in just a moment. So the other typical detection method is probes, and this is an example of attackman or hydrolysis probe. The probe has to be designed specifically for your target to hybridize between the forward and reverse primers. During the annealing step, the probe will hybridize to one of your strands. And because the reporter and quencher are held in close proximity, there's very low fluorescence signal. However, during the extension, the polymerase comes along as it's extending and cleaves the reporter from this probe. So now the reporter's free in solution and able to fluoresce. So we can detect this. So at the end of every cycle, as we're doubling our products, we're also creating double the fluorescence potentially. This is what we're going to detect. And then I mentioned before, I'll get to that point in a minute, about multiplex PCR. The instrumentation has evolved and gotten smaller, I guess you could say, so that we can have multiple excitation and emission filters in our optical module so we can detect multiple different colors. And so we talked about multiplex PCR last time, where we could have 4 individual reactions, or if we can make sure that these are designed to have different sizes, we could combine them into one reaction and then run a gel, saving sample and then saving space on our gel as well. But if we think about this and we carefully, again, have carefully designed primers, we design the probes now with different fluorophores, and we can look at this all in one tube. And this is an example of a 5-plex reaction in one tube. So that's interesting, but how can we use this information of the CQ? One thing we do routinely is use standard curves to monitor the efficiency of our reaction. We want to make sure that our primers are working properly and not causing primer dimer problems, and amplifying somewhere between 90 and 110%. That's our preferred range. So here's an example of a 10-fold dilution series. This is my most concentrated sample, and then each one is 10-fold lower. We can create a standard curve from this. Even if we don't have the exact input of our exact quantities, we know that they're all 10-fold dilutions. So we can create a standard curve and get a slope. In this case, it's minus 3.375. If we plug that into this efficiency equation, the efficiency we get is 97.8%. That's good. We're going to accept that. If you have a slope that's very different, you'll obviously get into a range that we don't want to be at. If you have efficiencies greater than 110%, you might have a primer dimer problem. So sometimes we do have to optimize reactions and change our anneal extend temperatures. So a 100% efficient reaction has a slope of minus 3.322, and therefore a 10-fold dilution series should have 3.32 CQ between each dilution. And a lot of times we can eyeball that and say whether our dilutions look good or not, but then we use the software. qPCR is often used for relative gene expression. In order to do this, we need to first screen reference genes to make sure that we're going to use a reference gene that's stably expressed in all of our samples. This is an example from a screen that I did with prostate cancer samples, and I wanted to find a reference gene that would be suitable to compare gene expression differences in these samples. The ones in green were calculated to be stable. The ones in red are not stable, so they are changing in different samples, and we wouldn't want to use that, as that'll introduce some errors into our analysis. The reference genes can be used to kind of to normalize for our input, sample input, once we've determined that they're stably expressed throughout our experimental samples. This is an example of relative gene expression where I first took one gene, reference gene, HPRT1 in this case, and we calculate the delta CQ, and then I have set the normal, which really should be called healthy, to my control. And here's an example, simplified delta delta CQ, where in each sample, we're going to get a CQ for our gene of interest, and we'll also get a CQ for our reference gene. This is a very simple one where the reference gene happens to be the same in all of them. But we'll think about the case where reference gene is varying a little bit, and that's why you'll see how important it is to know that it's stable before you use it. So we'll calculate a delta CQ, subtracting out the CQ of the reference gene from our gene of interest for each of these samples. Then we'll set one of the samples as a control. In this case, sample 1 is going to be our control, and we're going to take the CQ of our test, the delta CQ of our test, and subtract the delta CQ of our control. So all of these will be delta CQ minus 6. Here's our delta delta CQ. And then we can look at the relative value by expressing that as 2 to the power of negative delta delta CQ. So our control sample has a relative value of 1. And then these samples with differing CQs have different relative values. And that's basically how we would analyze our data. This is a very simplified method. There are more challenging methods that include multiple reference genes and also take into the account the efficiency of the reactions. So I've showed you a few examples of where I used qPCR to look at differential gene expression. It's also used in pathogen detection, environmental studies, screening for small RNAs, lung non-coding RNAs, etc. And happy to take any questions.
Interesting.
Stop sharing.
So I know I should know this, and I did at one time. I'm kind of curious, what is the sensitivity of the DNA binding dyes? You know, I know about ethidium bromide, of course, and cyber green. What was the other one? Eva green?
Eva green is one too. Yeah.
So why is that being used? I'm just curious.
Eva, I'm not sure exactly why Eva is. I think Evan probably has to answer.
Evergreen's a little brighter than cyber. It binds a little differently too. I want to say it binds less tightly than the cyber, and so it's less inhibitory. So we have some mixes where we've used cyber dyes, and we have others where we've used the evergreen dye.
Okay, thanks. Other questions?
I see one in chat about what CQs
Jacqueline.
what CQs have gotten with negative controls using cyber. The CQs should be 0, no CQ detected. If you have a CQ showing up earlier than cycle of 40, you have a problem with the sample. So if you have primer dimers, you will get a CQ. And that's part of the problem with reactions that have primer dimers, is that you can't really accurately quantitate because those primer dimers are part of the total fluorescence of the sample. Yeah, we went to attackman. Yeah, that's one option. Another option is to run a gradient PCR. We didn't talk about that even, but that's where you can look at different annealing temperatures for the primers to try to get a more specific reaction and maybe eliminate primer dimers that way.
So I guess people could talk too. They don't have to just put it all in chat. Well, I'm kind of curious, what is a gradient PCR then?
There are cyclers that actually have different temperatures on the different rows. So if you think of a 96 well plate, you can have the rows in the back are hotter and the rows in the front are cooler. And then there's a gradient in between. You can set the gradient, the range of temperatures.
Well, there is another question. Daniel asked, can touchdown PCR be used in qPCR?
I think it could. I haven't.
I worry that if you've if the PCR efficiency is not consistent through all of the cycles, that might mess with your quantitation.
Yeah.
If you were touching down, changing the protocol through your cycles. I haven't done PCR with qPCR this way, but
No, it might really depend on what's the question you're trying to answer. So if you're just trying to find out if something might be there, maybe touchdown PCR would be okay. I don't know.
Those of us
Okay.
who don't know what's touchdown PCR.
Yeah, I was just going to ask that, no, you can see I don't always get into the lab. So yeah, what is touchdown PCR?
Let me think. So with touchdown PCR, we're changing the temperature.
temperature. You
With
start out
touchdown,
high.
with touchdown PCR, you start at a little bit higher annealing temperature, and then you bring it down slowly so that things your primers might be binding to and causing problems may not cause problem. If you can get a little amplification of the real product, then it will go well after that. I was just thinking if you had a primer dimer problem, that maybe that would help to avoid that. But I can see how it would screw up quantitation too.
Yeah, I think if you had primer dimers though, you still have primers in the later cycles at lower temperatures and you'd still probably have them there. People had talked before about looking at the melt curve and the area under the curve to try to subtract away the influence of the primer dimers, but I'm not sure that that ever really was valid. Older, less expensive.
Do you see that, Elizabeth? Phil was asking, anyone, are there older, less expensive conventional PCR instruments available for educational purposes?
So I can take that one. Bio-Rad still makes conventional PCR instruments. That's something that we still do a lot. So there's various grades of instrumentation, and there's specifically educational PCR instruments available too as well. So there's all different varieties. Last week, no, 2 weeks ago, Liz talked a little bit about the conventional PCR market and showed examples of those instruments as well. So that one is available on video. I don't think you were there then, Phil, because I would have recognized you, your face.
One thing I was kind of curious about is how many people have a problem with the fact that RNA is very sensitive to degradation. And what are some of the precautions that people take to ensure that doesn't occur? Or maybe people don't have that problem.
I guess in my hands, I haven't really seen that problem. I always was taught to be afraid of RNA, be very careful, don't even look at it wrong or something, or it will degrade. But I haven't actually experienced that. in the lab. So I'm relatively clean at my bench. I try to keep my pipetters clean. I use sterile water. You know, I don't leave solutions out on my bench open. And I haven't had a problem with degradation.
Ah, so you don't do it in a hood or anything like that?
No. We did way back, you know, when we first started. But I just do it on my bench now.
Oh, wait, and this is another question. Jacqueline, do you want to say it out loud, or I'll read it? If you are trying to detect a pathogen at a very low titer level, will that exacerbate a primer dimer problem?
It could very well mask the detection, or you would probably get a positive result where it may or may not actually be there.
And is that an instance where you might turn to droplet digital instead of qPCR? Very low detection is one of the real strengths of droplet digital PCR over qPCR. There's kind of the different places where you use these different technologies.
Or you might try to use attackman assay, like Steve's going to talk about, I think, because that adds another layer of specificity. So you've got a third primer that has to bind to your sample.
And to Anne Halley's question, on next week, Linnea, Betsy Bodecker, and Lee Brown are going to be talking about teaching advanced PCR, and I'm sure they'll hit on, if you don't have an instrument, what can you do? So I'm sure they will be helping with that stuff.
Yeah, I just wanted to add on to that. I was teaching to some of the upper division students, and the students had struggled a lot. So I had to just give them a data set and help them understand what CT is and how to calculate the CT values and make meaningful conclusions about gene expression analysis. But it's a very tough topic to teach online. So that's where I was coming from.
All right. So let's move on. I would like to introduce Dr. Evan Bress. So you've already heard him speak today. So Evan is going to talk about his work at Bio-Rad. So Liz gave us an introduction, and thanks for that great introduction, Liz. And now we're going to hear about actual applications and uses in industrial laboratories. So Evan manages the tech transfer team in the life science group at Bio-Rad. His team is responsible for moving products out of the R&D phase, so out of research and development, and into the manufacturing group. Evan and his team develop production procedures for manufacturing, quality control test procedures for quantitative PCR, for cDNA synthesis, and for next generation sequencing products. Thanks a lot, Evan.
Sure. You've already heard me speak. Sorry, I do talk a lot. All right.
We love hearing you speak, Evan.
Yeah.
You've got a lot in that brain.
My mom once asked me if my parents let me talk at home. Okay, there we go. You can see my slide, right?
Yes.
Very good. So thanks for the introduction. My name's Evan Bress. I've been at Bio-Rad for just a little over 15 years now. And several weeks ago, I was approached, asked if I would say a few words about how I use qPCR in my my job. And aside from the obvious, which is I have a difficult time saying no, the first thing, well, the question I guess I asked myself was, why me, right? Why are you asking me? Because the truth of the matter is I'm not a typical qPCR user. So I'm not a molecular biologist. I've never been a molecular biologist. I'm like kind of a misplaced structure, protein structure guy. And so I'm not in the lab making, interesting scientific discoveries. I'm not like Steve, you'll hear about him in a moment. I'm not in the lab designing new and interesting products. I did that for about 5 years, my first 5 years at Bio-Rad. But for the last 10 years, as Bryony said, I've been managing this little group inside of R&D. We call ourselves Tech Transfer, and our job is to move product out of R&D and over into manufacturing. And so my focus really is on ensuring that the products that we make are going to serve our customers well. So to that end, I focus a lot on production procedures, and then we draft those and we train our production team on how to build the new product. My team spends a lot of time thinking about quality control testing. And I guess when I think about that, quality control testing, okay, I'm not a typical qPCR user, but I do use qPCR either in quality control testing, either for the purpose, say, of testing a new batch of a qPCR product, right? Or sometimes we use qPCR for quality control testing of our other products. So I thought there might be some interest today in me speaking a little bit about how we go about designing our quality control test procedures. And if I were to think about the challenges associated with QC testing and maybe try to summarize it in one word, that word would be uncertainty. All right. So move that out of the way. Sure. All right. If I could move it out of the way, you can help me. Otherwise, I guess I'll just leave it there. So there's a lot of uncertainty in the QC testing, and some of that actually has to do with, I'm not really sure how our customers are going to use our products. So most of the products, the vast majority of the products that I work on are not FDA regulated products. So if you're developing an FDA regulated product, you get to tell the customer exactly how they're going to use it. Okay, here are the samples you're going to run. Here are the instruments you're going to use. This is exactly the procedure you're going to follow, and you're going to do this every single time. For research use only products, which most of mine are, I don't have that, I guess, luxury, if you will. So I'm not entirely certain how our customers are going to use our products. And I don't want to overstate that, right? I know they're going to do qPCR, but I'm not sure, for instance, are they going to do gene detection experiments, right? Maybe they've got a cell culture and they've knocked out a gene using CRISPR, and they just want to see, is that gene there or not? So they might use qPCR for that, just to detect the presence of a gene. Or maybe they know that the gene is in there, but they don't know, they're interested to see, which alleles are there, right? Is it homozygous allele 1, homozygous allele 2, or some sort of heterozygote, right? So they might do some sort of genotyping experiment, looking at single nucleotide polymorphisms, for instance. Maybe they're interested, actually, I should have stole the other figure, Liz. I stole the figure right next to the one Liz used when she talked about gene expression. Maybe they're interested in gene expression. Maybe they've got some cells they've treated and they've harvested the RNA out of those cells. They've run it through a cDNA synthesis kit. So they've turned the RNA, copied the RNA into DNA, and then they use qPCR to quantify that DNA. In other words, to look at the levels of messenger RNA inside of that cell. Maybe that's what they're doing. I'm never entirely sure how our customers are going to use our products. And that leads to some uncertainty when I think about how to QC test it. In addition to how they're going to use it, so I'm never exactly sure what pieces of DNA our customers are going to amplify. We refer to that in PCR, right? You have a primer anneal on one strand and a little further down on the opposite strand, and that defines the region that's going to be amplified in the PCR reaction. Well, there's some rules when you should follow when you design an amplicon, and maybe not rules. It's more like Pirates of the Caribbean. It's like more of a guideline than a rule. But one of those guidelines might be you want the GC content to be right around 50%. And what I mean by that is that if you were to look at the sequence that you're amplifying and count the numbers of guanine and cytosines in there, about half of the amplicon should be G's and C's. And then, of course, by definition, the other half is A's and T's. That's a good rule of thumb when you're designing an amplicon. You'd like it to be in there. And the reason you like that is that G's and C's, everybody gets stacking energy, right? All the bases stack together when they're in the helix, but the G's and C's have 3 hydrogen bonds joining them together, and the A's and T's only have 2 hydrogen bonds joining them together. And so G's and C's are more difficult to break apart. It takes more energy. And so in that denaturing step of the PCR cycling, it becomes a little more difficult to pull the strand apart if you've got a lot of G's and C's than if you have, say, a lot of A's and T's. So you want to kind of find a balance in there. The amplicon length is also important. You want to make sure that you're not challenging, that your amplicon's not so long that the DNA polymerase has trouble getting through it. For qPCR, for PCR it's different, but for quantitative PCR, ideally you'd like the amplicon to be, I like right around 90 bases. So anywhere from 75 to like 100 bases. And you could go longer, you go like 200 bases or something. But typically you'd like the amplicon length to be just kind of in that nice, small, easy to work with region. Anything that makes it easier for the qPCR mix to do its job is going to make your quantitation better. And then there's primer design, and Liz alluded to this already as well. So the PCR primers are designed to anneal in specific parts of the DNA that's in your sample, right? And that guides what pieces are going to be amplified, the edges of your amplicon, if you will, right? Well, you want those PCR primers to anneal specifically at the desired spot. You don't want them all over the place because you end up getting non-specific products formed. Those PCR primers define the region that's being amplified. So if they're all over the place, then you have all sorts of products being amplified and you're not really quantitating that thing you want to quantify. And then likewise with primer dimers, you want those PCR primers to anneal on the DNA and not be so interested in each other that they stick to each other and you form these little primer dimers, which really act to poison the qPCR reaction because they either make it look like there's a signal that's not really there, or they use up all the PCR resources and affect your quantitation that way. So there's some guidelines, and customers, we hope that they would follow that, but they honestly don't always have the option of that. Sometimes the piece of DNA that they need to amplify, they need to look at, is in a particular spot on the genome. And the size is what the size is, and the GC content is what it is. And they don't have a lot of flexibility in terms of their PCR primer design. So, so they're just kind of forced into, here's what I have to do. And we want for our mix, our qPCR mix, to be able to do that for our customers. So when R&D designs a product, they really try to consider all of those uncertainties. How's a customer going to use it? What sort of amplicons might they throw at this particular product? And they design around that. When my team develops the quality control tests, we also try to consider those uncertainties. We think about that when we're designing the test, but the truth of the matter is there's no way that we could test for every possible customer application, right? So any of the applications, all the amplicons and so on and so forth, there's just, you don't have enough time to do anything like that. It would be impossible. But what we can do, and in fact, what we often do, is we'll select an amplicon that's not ideal, that doesn't fall within that perfect design space. Maybe the GC content's a little too high. Maybe it's a little on the longer side. Maybe the primers are prone to making non-specific products. And we'll do that to sort of stress the product in a way that it has been designed to deal with, but in a way that'll give us an idea about how we could expect that batch to perform for the customer. Another thing we do during development as well is we'll actually intentionally make broken products. So we know the formulation, you know, this is how to make the product, and we'll intentionally make a product that we know is going to work poorly. And then we can use that to evaluate the suitability of the various amplicons that we might be looking at for our QC testing. So we have some choices to make up front. What sort of amplicons are we going to select to test this product? And we make those choices and try to address the uncertainties around customer usage. But there's a third kind of uncertainty, and that has to do with measurement uncertainty. And we spend a ton of time thinking about this. So the ideal QC test is going to be reproducible. And in this context, what I mean by reproducibility is that if I take a sample from a batch and I run my QC test on it over and over and over again, I want to get the same answer every time. And that seems like a given, right? Well, of course you do. And it might seem like all you really have to do is run a few qPCR reactions, measure the CQ values, the CT, CQ, it's all the same thing. Measure those values, and you see what you get and determine whether your product's working. The truth of the matter is, though, quality control tests have to be carefully designed in order to make them reproducible. So there are a lot of factors that'll mess with you. The operator, for instance, whether that's a human operator or a robot, and we use both, the operator can have an impact on the reproducibility of your test. The equipment, maybe your pipettes, or recently pipette tips, believe it or not, instruments that go into the testing, that can have an impact on reproducibility. The quality and quantity of the DNA template that you're feeding into the PCR reactions, the quality and quantity of your primers and probes, if you're using probes, that can have an impact on reproducibility. And then there's what I tried to say here is like normal well-to-well variation. What I mean by that is, as Liz described the PCR process this week and last week, every PCR cycle you expect to perfectly copy all of the template in there. So you go from 1 copy to 2 copies to 4 copies and so on and so forth. There's a perfect PCR efficiency, right? Well, in reality, you don't always see perfect efficiency from cycle to cycle to cycle. This time in this particular well, only 97% of those copies got copied, or those templates got copied. And over in this well, it was only 95%, and so on. And the more you cycle, the more those little changes from well to well over each cycle will add up and will cause noise in your results. So that, and oftentimes you see it, oftentimes anything that comes up like around cycle 10 is super nice and tight. And anything that comes up around cycle 35, it's just normal variation. You're going to see a spread in the CQ values that you measure. So all of this, and combining the fact that we have our non-ideal Amplicons, means that we sometimes have a fair amount of uncertainty in the CQ values that we're measuring. And so how do you deal with that uncertainty? You can try to kind of impose some controls on your process. And in fact, we do that. We spend some time trying to control the process as best as possible to try to reduce the amount of variation that we see. But at some point, you can only improve it so much, and the noise is what the noise is. And so what do you do about that? The answer, of course, is you make repeat measurements. So if you know that you don't trust a single measurement terribly much, then you'll make multiple measurements, 2 measurements, 3 measurements, 4 measurements. That all sort of begs the question, how many measurements should you make? And the answer, I don't know, you might find it a little frustrating, is it depends, okay? How many repeats do you need? It depends. It depends on the precision of your individual measurements. So if you're making individual measurements and those CQ values are just coming in just bang, 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 right on top of each other, consistently over multiple experiments, then you probably don't need a lot of replicates. On the other hand, or rather, it also depends on the level of accuracy and precision that are required. So for instance, if you need to know, you need to measure just super tightly and super accurately, you might need a lot more replicates for that scenario than if, honestly, you don't need to have a very good estimate for that particular metric. So it depends on the level of accuracy and precision you need as to how many replicates you might need to just adequately describe the performance of the product. And that's really what we're after, is can we have some confidence that the product is going to perform the way we think it should for the customer, the way we promised it. It also depends on what you're measuring. So for instance, if you're measuring an absolute quantity, you throw 100 nanograms of DNA in, and then you measure some CQ value coming out, you might need some number of replicates for that. But if you have 2 batches that you've thrown into the test, say your test batch and a gold standard batch, and you fed them both, say 100 nanograms of DNA again, and you've got a bunch of CQ values. Now you've got these 2 populations. You might want to ask yourself, is the test batch and the control batch, do those look like they're performing the same, or is there a difference between them? If you're asking that kind of question, that'll have an impact on the number of replicates that you'll need to run. So we think about when we think about how many repeats, we spend a fair amount of time thinking about what kind of levels of accuracy and precision we need, what kinds of measurements are we making, and then of course the precision in the individual measurements. When we report out values, I really hate taking a bunch of numbers and crunching them down and reporting out an average. You take all this data and you turn it into a single number, and I feel like you lose a lot of information. So we tend to report our results out as confidence. And one of the reasons I like to do that is because it allows us to specify the accuracy of our reported results, right? So for instance, we might, here's a standard curve similar to what Liz showed earlier, and it's these are the fluorescence traces coming up. They're in log view rather than the linear view, which is why they look a little different here. But you could still see them crossing this threshold line. And these guys here at 100 nanograms are crossing that line. Just, let me do this, sorry. Just right around 13-ish cycles or so. You can see on the plot that they're all clustered together really nicely and tightly. We could just report an average CQ, but my preference is to take the number of replicates and the CQ values that we got and generate a confidence interval, so that instead of saying the CQ is 13.85, I can say, you know, I have 95% confidence that the true value for the CQ when I put 100 nanograms into here was between 13.8 cycles and 13.9 cycles. We do the same for PCR efficiency. Liz mentioned how the PCR efficiency is related to the slope of this plot of CQ value versus the log of the DNA concentration. Rather than just fitting a line and reporting back an efficiency of, let's say in this case, 92.6, the preference is to say with 95% confidence, and I believe that the true value of that efficiency is somewhere between 91.5 and 93.7. So this allows us, the confidence interval allows us to specify the accuracy that we're reporting out. And it also, the width of that confidence interval gives us a sense of kind of how noisy the data is. We can also design our experiments to try to control the width of that confidence interval. So kind of control the precision of the values that we're reporting out. So the first, after we select the Amplicon and the DNA concentrations that we're going to use and so on, our first step is to try to understand the variation in the system. And so what we'll do is we'll set up a whole bunch of identical qPCR reactions. Sometimes as much as like a full 96-well plate. We'll call that a uniformity plate. And we'll run those reactions on a bunch of different instruments. The intent of measuring the CQ value, getting as many CQ values as possible to try to get the best estimation possible for the standard deviation. In other words, how much noise is there in our data when we run this kind of qPCR reaction, this amplicon, this DNA concentration. And if we have that standard deviation, we can then begin to estimate how many replicates we need in order to get the level of accuracy and precision that we're looking for. So there are software tools that are available, a lot of software tools that are available. I use some old Excel spreadsheet tools that I got in some training years and years ago. But other programs like Jump or Minitab will help you with this as well. I think you can find this kind of thing online as well. Once I know the standard deviation, I know kind of the noise in my system, I can specify the confidence level, my accuracy, and I can specify kind of the precision that I want. So for instance, in this particular example, I know that these reactions are going to have a standard deviation of like around 0.2 cycles. If I'm willing to live with a precision of like 1 cycle, in other words, okay, I put this much DNA in, and I'm happy knowing that, you know, with 95% confidence, the CQ is somewhere within this 1 cycle range. Turns out I only need to run 1 sample. Now, I would never do that, but this is what the tool tells you. Now, as you want to tighten that confidence interval down, in other words, kind of shrink down that region. So you say, well, 95% confident, not in this region, but in here or even in here, that what that ends up doing is bumping the number of replicates that you need, increasing the number of replicates you need. So for instance, if you go from a half interval width of 0.5 to a half interval width that's about the same as the standard deviation, or is the same as the standard deviation, then you need four samples. And if as your interval gets tighter and tighter and tighter, for instance, in this case, now the standard deviation and the range are the same value, then you need a lot more samples here, 16 in this case. So we use the noise that we've measured, the standard deviation that we've estimated, and then we consider what sort of accuracy and precision we need. And we use that to tell us how many replicates we need in order to run the test. And I'm going to stop on this slide. I just wanted to maybe leave you with, it's not just about measuring absolute values like that. There are tools that one can use if you're trying to measure the difference between two things. In other words, the test batch and your gold standard batch. If you measure the standard deviation of your reactions, you know that. If you know the size of the difference that you'd like to be able to detect between those two populations, and if you establish the statistical power, which is the probability that you're going to avoid a false pass, and the confidence level, the probability that you're going to avoid a false fail, plug all that in, and you can estimate the number of samples that you would need to detect that size with these parameters given that standard deviation. And likewise, if you wanted to measure something that's like whether something is present or not, for instance, you could use a binomial tool to do that and estimate what the true positive rate might be. And there are times that we do that as well. So that is all I have to say for the moment. I hope perhaps that this has been a little helpful as far as maybe getting some insight into how we think about the quality control tests, but also really I feel like this applies just generically to experimental design. And I thank you for your attention, and I'm happy to welcome any comments or questions.
Wow, thank you. That's what I was kind of curious. Having done quite a bit of enzymology, much more so than qPCR, is this accepted in terms of research that depends on qPCR? I would think this type of quality control would be demanded of individuals who are using this technique.
I'm not sure I follow the question. Sorry.
Well, I mean, it's like your quality control is excellent. I would think that would be applicable to also what researchers are doing using qPCR.
Yeah,
Linnea, this is what they should be doing. It's not what they are doing. Do you know what the Mikey guidelines are?
I am passingly familiar with them. Go ahead.
Sorry, I was asking Linnea, but for publication, and Mikey is MIQE, and I forget what it stands for right now, but it's the minimum acceptable standards at the bench, at the design phase, at the execution phase for qPCR to be published. And some people are doing it, some people aren't doing it.
Lori, do you have a good reference for that?
Yeah.
Stephen Buston, I'll look it up. I'll put it in the chat.
That would be perfect, because I think it's important for students to know this and programs who are teaching this.
Yeah.
Any other questions for Erin before we move on to Steve? Evan, sorry.
That's all right. I answered a lot.
Talked about 3 Erins already today. There's lots of Erins in my life.
Evan, this was really excellent.
Oh, well, thank you. I appreciate that. We put a lot of, you can tell we put a lot of energy into.
You know, if I was a researcher, I think I would be picking your brain and when I was troubleshooting some of my research.
Well,
now we'll go on to Steve, who maybe sometimes does pick Evan's brain.
Yeah.
So Dr. Steve Aquino and his team at Bio-Rad developed PCR and DNA sequencing reagents for the life science group. Most recently, they've been working to develop reagents and diagnostic tests to help address the COVID-19 pandemic. And that's what Steve will be talking to us about today. So welcome, Steve. Thank you very much for coming to do this talk.
Thank you, and I'm really glad to be here. Let's see, I'll share my screen right now. Okay, my name is Stephen Okino. I'm a scientist at Bio-Rad Laboratories. I've been there a little bit over 12 years, and I develop products for the gene expression and genomics research markets.
Hey, Steve, we're not seeing your screen.
You don't see the screen?
No.
Oh, let me see if I could fix that. Is this better? Can you see the screen now?
Yeah, there you go, Steve.
Yep. Great, thank you. Okay, anyway, I was saying that, I primarily develop products for PCR and next generation sequencing. However, during the last year and a half, me and many of the colleagues at Bio-Rad, we've, I think, rather appropriately shifted our focus to develop products to address the COVID-19 pandemic. We first released kits for the diagnostic testing of COVID patient samples, but then later as the pandemic progressed, COVID mutations led to very dangerous COVID variants, and we became very interested in developing PCR-based COVID mutation assays, which can identify the exact variant a patient is infected with, and also help in COVID epidemiology and surveillance. So what I'm going to show you today is how what these PCR-based COVID mutation assays are, how they work, and I'm going to show you actually how to design them also. So first of all, so this is the background. So SARS-CoV-2, it's the virus responsible for COVID-19. The original SARS-CoV-2 virus, it's mutated into more dangerous variants. They're more infectious and can evade the immune response. The mutation in the SARS-CoV-2 virus genome, it can change the virus, increase its fitness, and each variant has a signature pattern of mutations that can be used to identify it. And by these variants, I'm talking about, things that are more dangerous, like the one that's shown up recently is the Delta variant, which is much worse than the original Wuhan variant. Currently, however, most mutation detection, it's accomplished by sequencing. What we've done at Bio-Rad is we've developed PCR-based mutation detection assays, and they offer several advantages. And I'm going to just give a brief introduction to what the advantages are. This gives a very, I guess, simplified view of what sequencing is. What they do when they want to look at, get the sequence of a SARS-CoV-2 virus in a patient sample, they start with the patient sample. They then perform many, many steps to make a sequencing library. They sequence it, analyze the data, and after about 2 or 3 days, they have data on the, genomic sequence of the virus. However, sequencing, it's really expensive, it's time consuming, and very technically challenging. It's not performed in SARS-CoV-2 testing labs where they actually, are able to make diagnoses of if patients are infected or not. Because of this, most SARS-CoV-2 samples are not sequenced, and there can be a significant time delay between collecting a sample and posting the sequence results, especially in third world countries where they don't do a lot of sequence, they don't have access. So therefore, worldwide variant surveillance, it's spotty, slow, and the fact that it relies on sequencing really contributes to this issue. So this is the PCR workflow that we developed. You could start with a patient sample. You then have a mutation assay, which I'm going to talk about in a little bit, and also a PCR reagent. You just mix these things, throw it on a PCR plate, and then a real-time PCR instrument. And in about 90 minutes, you have your data. You could tell if a mutation or what mutations, the ones that you're going after, if that mutation is present in the virus sample or not. The PCR-based mutation detection is very fast and easy. It's identical to SARS-CoV-2 testing, and it can be accomplished in SARS-CoV-2 test labs. Therefore, implementing PCR-based mutation detection can improve the coverage and speed of variant surveillance worldwide. So how does this work? How does PCR work to identify the mutations? And the answer is that we use a very specialized type of PCR. It's probe PCR, the ones that look for specific mutations. Here I'm showing the advantage of an example of the E484K single mutation assay. So what this does is it looks for a mutation in the 484th codon of the SARS-CoV-2 spike gene, which changes the E, which is glutamic acid, to a K residue. It turns out that this mutation is very important. It's arisen independently in several variant strains, and it makes the virus better able to resist the immune response. This assay and all single mutation assays we developed, they have a forward primer, a reverse primer, and 2 probes. There's a wild type probe. It's labeled with a hex dye, and it has a wild type G in it. So this is, the G is the base that is consistent with the Wuhan. It's in the Wuhan reference gene or virus. It also has a mutant probe. It's labeled with FAM, so a different fluorophore. And this one here has an A instead of the G, and it targets the mutant virus. The way this assay works is if a wild type template like the Wuhan virus is present, well, and you run this assay with it, what you'll see is that you'll see a hex signal because this wild type probe will bind there. The mutant probe will not because it does not have an A there. And because it's binding, you'll see hex signal. However, if you're looking at a different mutation like the beta variant, which does have the mutation of interest, here you can see that the FAM probe is binding, the hex probe is not, and the mutant probe will produce a FAM signal. There's an other choice here, a third possibility. In some cases, different variants arose that have been mutated in the same position, but with a different base. For example, the kappa variant, it has mutation at the E484K base, but the mutation is a C rather than the A found in the beta variant. This mutation causes an E to Q amino acid change. And in this case, both of the probes in this assay will not bind. They are not perfect assays and no signal will be emitted. So a single mutation assay could give 3 types of calls: wild type, mutant, or no calls, which means the virus that you're looking at, it's not wild type or mutant, it's different. These are the results of the 484K assay, what you're looking at. When this assay analyzed the Wuhan virus, you can see that you have very high hex signal here, which is what you would expect because it's wild type. However, when you're analyzing the beta or the beta variant, you see very high FAM signal. And when you analyze the kappa variant, you get no signal. The signal, in the qPCR trace, it's consistent with the no template control. You could do an auto analysis in some software that Bio-Rad developed. It'll take this type of chart here and throw it, using allelic discrimination results and automatically call what the mutation is. So here it's automatically calling the wild type virus. The wild type virus is Wuhan, and also correctly called the mutant beta virus, and it correctly called the kappa as a no call. So this allows you to make the mutation calls and make sure that they're correct. So this is what a mutation assay does. So now what I'm going to do is go into telling you about how to actually design them. So the requirements for designing this, the first thing you need a database that contains the genomic sequences of SARS-CoV-2 viruses. Here I'm using a website. This is free, okay? It's GISAID. They collect genome sequences of SARS-CoV-2 throughout the world, and they annotate everything and they put it there, and it's free to use for anybody. After that, you need a sequence aligner to align the genomes so you could identify the mutations. Again, here, this is a free web-based tool. I use Cluster Omega to align the sequences. And all these are free. You could just sign up and you're in. You also need a person to look at the aligned sequence and design the primers and probes for the PCR assay. You need synthetic nucleic acid corresponding to the wild type and variant sequences to make sure that the PCR assays work. And here we're just using a DNA synthesis company called IDT to do this. So this is what you see. This is Assay. This is their the free database of SARS-CoV-2 viruses. This is actually what a lot of the SARS-CoV-2 epidemiologists are using right now. And it's, I gotta say, it's really great, okay? So what I did here is I'm saying like, okay, suppose they want me to go after the Delta variant. You have this box over here where you could specify, I wanna go after Delta. So only the Delta, SARS-CoV-2 viruses corresponding to the Delta variant will be shown. I then said I wanted only ones that were submitted between 9/1 and 10/22. So it just, filters everything out. You only get viruses that were submitted during these dates. And then I said, okay, I want only things in North America. You could go over the whole world. It could be Asia, Europe, Africa, all other places, but you could choose exactly where you want to go. When you set these types of parameters, it turns out there's a total of 318,000 viruses. This is a list. You could see these are the different viruses. There's many, many pages of them. And then what you could do is you could just click on any one of these viruses over here. And this is what you'll get. So this is the details of that one specific virus that was entered into Assay. It gives you all the mutations over here. But most important, they give you a FASTA file that gives you the genome sequence, and that's over here. Typically, I'm collecting the FASTA files. Typically, I'll collect 12 to 15 genomes from different areas of the world are downloaded for each variant. So then the reason why different areas of the world, because you're looking for mutations, you want to make sure that the mutation is consistent with the variant and not just the region that it's in. I take a whole lot of these variant FASTA sequences. It could be one variant and then you add another variant. You need your controls in there. You just compile a lot of these FASTA sequences and you align them with Clustal Omega. This is the aligner that I'm using and that's shown here. So this is the Clustal Omega website. I'm doing multiple sequence alignments. And the tool, it could align up to 4,000 sequences at once. So this is, you just punch in your sequence. I put in the header here. So this is Delta from USA1, and you got the sequence here. And below that, you have many, many other variant sequences. You just say, go. And then typically in a couple of hours, you get, a message that, hey, everything's aligned, and you get an output file. Okay, so this is just one page of the output file. There could be hundreds, and I've actually gotten over 1,000 pages. But what it is just aligning the sequences of all the different things that you put in here. Here, what I did is the Wuhan human one, this is the wild type. I put in the spike gene because I'm going after only the spike gene. That's what I'm interested in. This is amino acid 1, 2, 3, etc. And what you could see here is that all the sequences are the same except this gamma variant, the gamma variant 3 that I grabbed, it missed all these things. So then this shows up, okay, the bases are not consistent with everything else. So those are wrong. So that's what you're looking for. You're looking for differences. Now here, suppose they said, okay, they want me to look at variant 1 and design an assay for this particular mutation here. You can see that there's a mutation here. It's different, the base is different than the wild type Wuhan virus. But then so this is variant 1, this is the target that we want. But then you can see that in variant 2, 3, 4, you have different mutations in different regions. And if you design your primers with with one of these mutations in them, it will not work. You really have to get around these. So to develop an assay for variant 1 mutations, the primers and probes cannot overlap mutations in other variants. And the other thing, I make 2 probes, one corresponding to the Wuhan sequence and one to the variant 1 sequence. This is an example now of an assay that I designed. Here you could see the two primers. You got they're in yellow. You could see what I was trying to do here was stay away from this T. So then I, just put this primer here so it does not overlap here. I would prefer to have this primer in the middle of these two, but then it did not, I guess, it just didn't work. The TM was too low, so I had to take it outside of there. These are the two probes that I designed. The wild type one has the T, the mutant has the C. So that's the assay that was designed. It's preferable to have the mutations that you're looking at be in the middle, near the middle of the probe. It's preferable for this assay to be as short as possible, but lots of times mutations and other variants will, make it so you can't do that. So you just have to sort of work around everything to the best of your ability. So once you design the assay, then you just really have to test it. So this is what we do. The target mutation assay, it's tested with wild type and variant nucleic acid, and I use both a low and a high concentration. These are the PCR traces. So you can see here, the variant high is in red, it's picking it up. Wild type high is green. This is like many wells on a plate. So there's some that I put, variant at high concentration, wild type at high concentration, variant at low concentration, which is out here. The CQ values are in the 35 range. Wild type at a low concentration. And also in this one, we have a sample control, which is consistent across. And if you do everything, you look at this data here, you put it through allelic discrimination, and you can see everything did cluster correctly. The wild type high and low were called as wild types, and the variants high and low were called correctly as variants. So for an assay to release these mutation calls, we need a high and a low. And actually we do other work to make sure it's not picking up something that's very similar. So this is the basics on how we'll QC an assay that we developed in order for it to be released to the public. Okay, once we develop, that's going after a single mutation. Afterwards, we were talking in the halls with just people in the lab. We did creative brainstorming and actually improved this mutation assay considerably. Now you can see we have just the two primers still, but we have 4 mutations that we're looking at, and also a positive and control, the MPC, which just is the hex probe. It's consistent with just the Wuhan virus, and it just makes sure that the SARS-CoV-2 virus is present. But here we could look at mutations at 4 critical SARS-CoV-2 sites in a single reaction. Also, this is a patent pending, this is assay, or this, I guess, technology, looking at multiple mutations all in one assay. It's patent pending. A lot of people in the lab, they've been doing PCR, even probe-based PCR for like 2 decades. And nobody's seen an assay like this, where you have just 2 primers and a whole bunch of probes in the middle. So that's where it's innovative. But that's where brainstorming and just talking with people can get you to improve things that you already have. So this is our summary. We developed these PCR-based mutation assays. They identify SARS-CoV-2 mutations. The assay to design, it involves development of a multi-resource pipeline, like just say, the aligner, everything. And this took a lot of trial and error and continuous learning. At first, it took like a couple of days to develop an assay. Now it probably takes an afternoon or so, not counting the time that the aligner takes. The other thing is that, working in industry, you think everything is, pretty much set, but no, a lot of this creativity and thinking outside of the box, it's really important to develop these innovative products. And that's sort of something that, I really like to see in people who are coming to industry, how open-minded they are, how they could see outside the box, and can they potentially develop something that's new and innovative. So with that, I'll stop and take any questions.
Wow, excellent. Here's one new message. Oh, very well explained.
Okay.
So what I was really curious about, of course, is they have identified hypervariable regions within the spike protein. So I wouldn't be surprised if these mutations are within those regions themselves. Not that.
Yes, sorry, I didn't mean to interrupt. The hypervariable region that's really important is this particular region in the spike gene that that I showed you in the multiple mutation assay. Although, all those mutations in there, they do make the variant a lot, I guess, more, increase its fitness, makes it dangerous. So that's why we're targeting that particular, hypervariable region, which makes the virus a lot stronger when mutated.
Well, this will be interesting. When the new coronavirus shows up, you'll be able to design assays immediately to detect variants.
Yes, in fact, in Bio-Rad, that's what we're doing. We'll, every month, we'll look at, what new variants are out there, what new mutations can be interesting to go after, and then design and release assays for them. So yes, this is a continuous project. It'll only end hopefully when the SARS-CoV-2 goes away or becomes endemic or something. But yes, this is something that is continuous.
I was wondering, would this have any implications for the flu too?
Okay, these mutation assays, yes, they will work on pretty much anything.
Any virus, right?
Yeah, any virus. And it's not even just viruses, it could work on anything. Okay. So then here we're going after these, SARS-CoV-2, it's an RNA virus. So we're doing something called RT qPCR here, where we're doing a cDNA synthesis step baked into the amplification and quantification, or I guess, probe detection. So yeah.
Wow.
So we're coming up to time right now. I would like to remind everyone, if you're not aware, the Bio-Rad does have a real-time PCR educational kit adaptation for developing a COVID-2 assay, a COVID assay. So not quite the technical level that Steve talked about here, but we do have an adaptation of our one of our real-time PCR kits that you could use to start to teach about this kind of work. Yolanda's just put it up in the chat. So thank you so much, Steve, for this. I think Purnima has some logistical questions for you. I will encourage you to do the survey. It's really important for the grant, and there is the opportunity to get a free Bio-Rad poster if you do complete the survey. So there is another reason to do that, not just to help Purnima and Linnea and Cassandra on future grant applications. Thank you. Thank you, Liz and Evan and Steve, for speaking today.
Thank you. Amazing. Thank you.
Oh, yeah, Laurie, question?
I'm not sure if it's a question or if I wanted to say something. So this was great, by the way, and I'm really sorry I was late. So several years ago, we've been teaching qPCR for a while, but what we used to do was focus on regular old PCR, and then qPCR was like, well, this is cool and we have one. But several years ago, I called it an exercise in precision pipetting. And some of the things Evan was showing is how we used it. We didn't care what the heck we were amplifying. The students made a standard curve. Really, the sole purpose was for them to look at their pipetting. They did the whole plate by hand, and to look at their pipetting and either go, hey, this is okay, or, oh my gosh, I thought I learned this and it looks like I have more practice I have to do. And if you're sort of just getting into it, I think that's actually a really good use for it. There weren't a lot of jobs where I am. I'm in Montgomery County, Maryland. And several years ago, people wanted it, but they weren't hiring dozens of people. Right now, anybody who can do RT-qPCR will have a job tomorrow because of all the COVID testing. So we've changed how we're teaching it, but depending on your area, some of those QC tests, don't think of them as like making your assay better, think of them as making your students better. And I don't know. Anyway, that's how we used to use it.
I actually think that, Stephen, it'd be really interesting to put out a challenge to the educational community to see how your assay could be applied to other pathogens.
That's a really good point. Yes, because right now this assay, especially that multiple mutation one, which is, a lot more powerful, we're only using it on COVID. Can it work in other scenarios? Yes, probably. Yeah, it would be great to ask the academic community, are they working on something that can be used, that this assay could be useful for? And I do think it potentially could help a lot of, I guess, areas of research. I just don't have enough background to know where, but yeah, throwing out to the academic community to ask for, suggestions could be very helpful. Thank you. That's a very good point.
Well, once again, I think these PCR webinars have turned out to be absolutely fantastic.
Agree. I'm only just a high school biotech teacher, guys, and just the amount of knowledge I'm getting from you two at Bio-Rad is like amazing. I'm just like learning curve is going up really well. Understanding all the different applications, the background comparisons. I love that comparison with the next gen sequencing. It's important for the students to go into college kind of knowing already how the different departments work together in a biotech company, and to, again, maximize what the product is capable of and the different ways that they can spin it. Great job, guys.
I concur. I'm also a biotech high school teacher, but I'm adjunct with a university. So this will bridge it nicely for my seniors right to freshman college. So I thank you both, Evan and Stephen. Well explained. And now I know what I need to do. Thank you so much.
I just want to add that these series, the very first part, which was a showcase, and the next one, which will be coming up next week, all the 3-part lecture series will all be available on InnovATEBIO website for you to go back and listen, which is what I'll be doing. I'll be listening to this over and over again. There's a lot of information here. So yes, it will be available for you to go check out on our website. Thank you so much.
Great, thanks everyone for sticking around. And if you have any more questions, please ask away. Otherwise, please fill out the survey. You can see the eagerness from Cassandra and Purnima. We really need that survey filled out, so please do that.
And Bryony will be sending it out by email too. So we're really gonna hound everyone.
Yeah. We're not done yet, but if you take the survey, there is an option to get a poster from Bio-Rad. So make sure you fill that part out as well.
Yeah.
All right. Thanks, everyone.
Thanks. Perfect.